So the next experiment that I am going to explain is cloning induction. This is somewhat a complicated multi-step experiment. So as usual, I am not going to detail the entire protocol. Step by step, one by one, we can continue, right? So now I'm going to explain the theory. This is the foot and mouth disease virus genome, right? This is FMDV virus genome. Our lab has identified a specific sequence called OVL that can induce an immunity in the animals, for example, cattle. Right? Didn't understand? This is the FMDV sequence, the entire genome, foot and mouth disease virus genome. So our lab has identified specific sequence, a fragment called OVM, just a name, that can potentially induce an immune response in the animals against the original virus. Right? Didn't understand?
Induce what?
Immune response.
Immune response.
Immunity.
Okay.
Right. So it means we have to inject, we have to vaccinate this animal. Right? So any foreign agent that can induce an immune response is called what? Foreign agent that can induce an immune response is called what?
뭐라고 하지?
We call antigen.
Okay.
Antigen. So when an antigen is injected, immunity is built in the animal. So when the actual virus comes, the animal can be safeguarded from the actual virus infection.
Okay.
Right?
Yeah.
So that's the principle. So our lab has developed a specific sequence called OVM from the foot and mouth disease virus that can potentially induce an immune response. Right? To check this one, we need to satisfy two things. Right? We want to know, once we inject this sequence, we don't know whether the OVM sequence produces a protein or not. So we want to confirm that once injected, this OVM produces a protein. That is the first objective. We want to know the protein is expressed or not. We want to check.
Okay.
The second objective is, so the OVM protein, should the OVM protein soluble or not? Soluble or not? What is your idea? I am asking whether the OVM protein should be soluble or not.
Soluble?
Why?
돼야 되는 거 아닌가?
Because the blood contains water,
Yes.
so our antigen should be soluble in the water.
Okay.
So therefore, we need to know soluble or not. The experiment that satisfies these two requirements called cloning induction. Do I need to explain again?